In this video, we are going to learn about the buffer solutions. A solution that resists change in pH upon addition of small amount of strong acid or a strong base is called a buffer solution. The capacity of a solution to resist change in its pH value is known as buffering capacity. Buffering capacity. Example for buffers are acetate buffer, phosphate buffer, citrate buffer, bicarbonate buffer. Which are the examples? Acetate buffer, phosphate buffer, citrate buffer, and bicarbonate buffer along with the acetate and citrate buffer. A buffer solution is an aqueous solution consisting of a mixture of a weak acid, there will be a weak acid, and its salt, or a weak base and its conjugate acid. This is actually something which is weak, not strong. A weak acid and its conjugate base, or like a weak acid and its salt, or a weak base and its conjugate acid. Its pH changes very little when a small amount of strong acid or base is added to it. Buffer solutions are used as a means of keeping pH at a nearly constant value in a wide variety of chemical applications. Many life forms thrive only in a relatively small pH change, so they utilize a buffer solution to maintain a constant pH. An example of buffer solution found in nature is the blood. Never forget the example of blood. When a drop of concentrated HCl added to a little of pure water, the pH of water changes immediately from 7 to 2.2. Similarly, if a drop of NaOH solution is added to a little of pure water, the pH of the water increases from 7 to 13. However, such changes will not be occurring in buffer solutions. Even when 1 ml of acid or alkali is added to the buffer solution, the pH of the solution will remain the same. The buffer solutions possess reserve acidity as well as reserve alkalinity. Thus, ammonium acetate has reserve acidity due to the presence of CH3COO minus ions and reserve alkalinity due to the presence of NH ions. Reserve acidity due to the presence of CH3COO minus and reserve alkalinity due to the presence of NH concentration. Any solution containing a weak acid together with one of its salts or a weak base with one of its salts functions as a buffer. The buffer may be an acid buffer or a base buffer. Acid buffer or a base buffer. An acid buffer consists of a weak acid and its salt. Example, CH3COOH plus CH3COONA. CH3COOH plus CH3COONA. Acetic acid and sodium acetate. A basic buffer is a mixture of weak base and its salt. Example, NH4OH plus NH4Cl. NH4OH plus NH4Cl. A buffer can be represented by placing the acid or base as numerator and their salts as the denominator. Like acid buffer is equal to acid by salt. Basic buffer is equal to base by salt. What is the example of an acid buffer? CH3COOH plus CH3CONA. What is the example of basic buffer? NH4OH plus NH4Cl. NH4OH plus NH4Cl. Mechanism of buffer action. The mechanism of action of acid buffer can be explained by taking a buffer solution of a weak acid, acetic acid, and its salt, sodium acetate, like NH4OH and NH4Cl. The dissociation of CH3COOH in the solution is very small, we are explaining as acid buffer, so we will be taking the example of acetic acid and salt of that acetic acid, like sodium acetate, CH3COOH and CH3CONA. So CH3COOH gives CH3COO minus plus H plus. This means that there are Very few H plus ions present in the solution. The presence of CH3COO minus ions in the solution further suppresses the ionization of CH3COONA, which is a weak electrolyte. So CH3COONA gives CH3COO minus plus Na plus. When a drop of HCl is added to the above solution, it reacts with the salt component of the buffer system. The acid immediately ionizes into H plus and Cl minus ions. The H plus ions combine with CH3COO minus ions to form CH3COOH. The Cl minus combine with Na plus ions to form NaCl. An acetic acid is not easily ionized. pH will not increase. Like HCl gives H plus plus Cl minus. So when we add this HCl into the CH3COONA, we will be transforming into CH3COO minus plus H plus. And H plus CH3COO minus gives CH3COOH, and N plus Cl, Na plus Cl gives NaCl. When an acid is added to the buffer, the salt component reacts with it. What is reacting with it? Salt component will be reacting with it. Similarly, when an alkali is added to a buffer, it reacts with the acid component of the buffer. When acid is added, it will react with the salt component. And when alkali is added, it reacts with the acid component. Sodium hydroxide and added into acetate buffer immediately ionizes into Na plus and OH minus ions. The OH minus ions combine with the H plus ions to form water. The Na plus ions combine with CH3COO minus ions to form CH3COONA. NaOH gives Na plus plus OH minus. CH3COOH transforms to CH3COO minus plus H plus, but H plus OH minus gives H2O. Along with that, Na plus plus H3COO minus gives CH3COONA. Acetate buffer. Next one we are reading with the acetate buffer. Acetate buffer is prepared by mixing acetic acid with sodium acetate solution. Prepare stock solutions of acetic acid and sodium acetate. Acetic acid stock solution mix 12 ml of acetic acid, 12 ml of acetic acid, and 988 ml of distilled water. Sodium acetate stock solution dissolved 27.2 gram of sodium acetate in 1000 ml distilled water. For preparing acetate buffer of desired pH, mix acetic acid and sodium acetate in the suitable proportion. Phosphate buffer. Prepare stock solutions of monosodium hydrogen phosphate, and disodium hydrogen phosphate and mix them. To obtain phosphate buffer of desired pH, mix the two stock solutions in the desired proportion. Then tris buffer is there. Prepare stock solution by dissolving 24.2 gram of tris. What is tris? Hydroxymethyl aminomethane. Hydroxymethyl aminomethane. Hydroxymethyl aminomethane. In 1000 ml distilled water to 25 ml tris stock solution, add 0.1 normal HCl as indicated and dilute to 100 ml with distilled water. So that is happening in the case of tris buffer. Now let's learn about the application of buffers. Buffers protect cells and tissues from sudden changes in pH. Buffer solutions are necessary to keep the correct pH of enzymes in many organisms to work. Many enzymes work only under very precise conditions. If the pH moves outside of a narrow range, the enzymes slow or stop working and can denature. In many cases, denaturation can permanently destroy their catalytic activity. A buffer of carbonic acid buffer and bicarbonate are present in blood plasma. Carbonic acid and bicarbonate are present in the blood plasma to maintain a pH between 7.35 and 7.45. Bicarbonate buffers help in the removal of CO2. Bicarbonate buffer helping the removal of CO2. What is the pH? 7.35 to 7.45 to maintain pH in the air level. Industrially, buffer solutions are used in fermentation process. Used in the fermentation process. And in setting the correct conditions for dyes used in coloring fabrics. First application was in the case of fermentation process. Next, in the application of dyes by providing correct conditions. They are also used in chemical analysis and calibration of pH meters. As we have mentioned earlier, it is used in the fermentation process and dye coloring, correct condition providing of dye coloring of fabrics, and in chemical analysis and calibration of pH meters. Phosphate buffer. The majority of biological samples that are used in the research are made in buffers, especially phosphate buffered saline PBS. Phosphate buffered saline at pH 7.4. It is the buffer solution commonly used in biological research. It is a water-based salt solution containing NaCl, Na2PO4, KCl and K2PO4. And buffer helps to maintain a constant pH. PBS is isotonic and non-toxic to cells. Isotonic and non-toxic. Phosphate buffered saline, pH of 7.4, and is used commonly in biological research. And this is a buffer with water and along with salt solution containing NaCl, Na2PO4, KCl and K2PO4. The buffer helps to maintain a constant pH. PBS. Phosphate buffered saline is isotonic and non-toxic to cells. It can be used to dilute substances. It is used to rinse containers containing cells. It is used as a diluent, used to dry biomolecules. It is used to rinse the container. Acetate buffer. It is used in textile industry to neutralize the sulfuric acid wastes as a photoresist while using aniline dyes. It is also used in chrome tanning in the synthesis of synthetic rubber. Sodium acetate is added to food as a seasoning and also used in the biochemical reactions. Tris buffer. Tris buffer is used in polyacrylamide gel electrophoresis. It is also used as an alternative to sodium bicarbonate in the treatment of acidosis. It has wide applications in cell culture and pharmaceutical industry. Tris buffer is used in the case of polyacrylamide gel electrophoresis. So, which are the main applications of buffer? Buffers protect cells and tissues and protect enzymes to preserve their catalytic activity and prevent them from denaturing. And a buffer of carbonic acid, bicarbonate are present in the blood. They maintain the blood pH between 7.35 to 7.45. Bicarbonate buffers help in the removal of blood CO2. Next, industrially speaking, they are used in fermentation process and used in dye coloring and chemical analysis and calibration of pH meter, along with the phosphate buffer in detail. Phosphate buffer is also called as a phosphate buffered saline, and it's used in research to maintain the pH of 7.4. Constant pH is maintained with this, and it's the isotonic one and non-toxic to cells. So it is helped to carry out the experiments and used it to dilute substances and to rinse the containers containing cells. Acetate buffer is also there. It's used to neutralize the sulfuric acid wastes in textile industry and used in chrome tanning and aniline dyes and synthetic rubber production. And This buffer is used in the polyacrylamide gel electrophoresis.